Estimation of hemoglobin concentration by cyan-meth hemoglobin method. There are several methods of estimating hemoglobins, such as hemoglobin color scale, Charlie's technique, use of HemoQ, and use of automated hematology analyzer, etc. However, cyan-meth hemoglobin method is the internationally recommended method for determining hemoglobin. Learning objective. The objective of this video is to appraise the students about estimation of hemoglobin by cyan-meth hemoglobin method. This video highlights principle, procedure, and results interpretation. Principle. Blood is diluted in the Drabkins solution containing potassium cyanide and potassium ferricyanide. There, the hemoglobin is oxidized to methemoglobin by potassium ferricyanide, which is then converted to cyanometh hemoglobin by potassium cyanide. The absorbance of the resulting solution is then measured in a spectrophotometer at a wavelength of 540 nanometer or in a colorimeter using a yellow-green filter. Equipment, materials and reagents. Equipment, micropipettes and a glass pipette, a spectrophotometer or colorimeter. The principle applied for the estimation of hemoglobin is the same in both. However, the wavelength available for the estimation differ. Materials. Alcohol wipes, cotton swabs, needles and syringes or lancets, EDTA tubes, and test tubes. Reagents. Drabkins solution. It is a mixture of potassium ferricyanide, potassium cyanide, potassium dihydrogen phosphate, Triton X, and distilled water. It is also available commercially. Drabkins solution is unstable if exposed to sunlight and can be stored at room temperature in brown borosilicate bottles for months. If room temperature is greater than 30 degrees of Celsius, store in refrigerator but brought to room temperature before use. The solution should be clear and pale yellow in color. pH of the solution should be between 7 to 7.4. Discard the solution if turbid and the pH of the solution is out of range. Hemoglobin standard. This is available commercially at a specific concentration. It is photosensitive, hence exposed to light causes deterioration of the strength of the standard. The concentration of the standard used in this practical is 15 grams per deciliter. Blood samples. Both EDTA blood collected by venipuncture or capillary blood by finger pricking can be used for this method. Procedure. In this demonstration, we use capillary blood collected by finger pricking. Clean the chosen finger with an alcohol wipe. Twist and break the tip of the lancet to open the needle. Prick the finger and squeeze slowly until the blood comes out. Wipe away the first drop of blood with a clean tissue. Draw 20 microliter blood into the pipette and wipe outer surface of the pipette with the tissue to remove excess blood. Place the pipette into the tube containing Drabkins reagent and slowly expel the blood into the solution. Mix well and stand for 15 minutes. Take 5 ml standard hemoglobin solution into another tube. Set the wavelength of the spectrophotometer to 540 nanometer. Blank the machine using Drabkins solution. Record the absorbance of the test sample at 540 nanometer. Measure the absorbance of the standard in the same manner using Drabkins solution as the reagent blank. Calculation of hemoglobin concentration. Hemoglobin concentration in the sample is calculated using this formula. Concentration of hemoglobin in the sample is equal to absorbance of the sample divided by absorbance of the standard and multiplied by the concentration of the standard. Advantages. All forms of hemoglobin except sulfur hemoglobin are converted to cyan-meth hemoglobin. Visual errors is eliminated as no color matching is required. A reliable and stable reference standards are available from WHO. Disadvantages. Potassium cyanide is highly poisonous. That is the reason for Drabkins solution never pipetted by mouth. Abnormal plasma proteins and high leukocyte count causes turbidity on dilution of blood in Drabkins solution, which can be overcome by centrifuging the diluted blood. According to WHO, these are the normal ranges of hemoglobin in adult males and females. However, these are arbitrarily defined values as it depends on several environmental, biological, and physiological factors such as high altitude, pregnancy, physical exercises, etc. If the hemoglobin level is below the normal range, the condition is referred as anemia. There are several reasons. It could be due to iron deficiency, red cell membrane defects, enzyme defects, bone marrow defects, etc. I hope that you would have gained a clear understanding on Drabkins method of hemoglobin estimation and its advantages and disadvantages. However, this classical method is not used routinely now in diagnosis of hemoglobin in modern laboratories. Automated analyzers are used to obtain full blood count in which you could get hemoglobin levels together with the other red cell parameters. Finally, I would like to thank Dr. Abhita Abegunasekara, Mr. Nuwan Wickramapada, and Mr. Susantha Senarath for supporting me to prepare this video. Thank you.